As you can see, the chromatid arms, one chromatid arm, the second one, third one, fourth one, they will form a tetrad chromosome. So if you remember, I had told you mitosis is a equational division. The same number of chromosomes will be carried to the daughter cells as well. Here recombination is occurring, recombining. A portion of the genes that were present here will get transferred to the neighboring chromosome. Hello everyone, a warm welcome to the last session of this chapter, that is cell cycle and cell division. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had concentrated on the M phase, that is the mitotic phase of cell division, wherein we studied the different stages under the mitotic phase, which was the prophase, the metaphase, the anaphase, the telophase, and finally the cytokinesis. In today's session, we shall completely concentrate on the meiotic cell division, and meiotic cell division usually occurs in the reproductive structures such as the gametes, wherein if it is a human being, it is the sperm, which is the male gamete, which is haploid. In that, meiotic cell division will occur, and in the case of female, the ova or the egg, the meiotic cell division will occur. And if we take the example of a plant, the pollen grain, in which the male gametes will be produced. So there also the meiotic division will occur. That is, in the pollen grain, the meiotic division occurs. So today we shall completely discuss about the different phases of the meiosis. So to begin with, we shall first understand what meiosis is. So meiosis here, it is a reduction division. Why is it a reduction division? So if you remember, I had told you mitosis is a equational division. The same number of chromosomes will be carried to the daughter cells as well. But in the case of meiosis, here the chromosomes will reduce in their number, will become half when compared to that of the parental cell. So meiosis is a kind of cell division that reduces the number of chromosome by half. Say for example, if a cell is dividing, here there are say 20 chromosomes, the chromosomes will be distributed to the daughter cells exactly half, that is 10 to one daughter cells, the other 10 chromosomes to the other daughter cells. So there is reduction occurring here, right? So therefore, it is called as the reduction division, which is nothing but meiosis. Here, the cell division reduces the chromosome number by half, which results in the haploid daughter cells. So, haploid daughter cells will get produced. So, from a diploid cell, haploid daughter cells will get produced. So, therefore, it is nothing but meiosis. So, meiosis makes sure that the production of haploid phase in the life cycle, that is usually meiosis is seen in the haploid phase in the life cycle of sexually reproducing organisms, wherein they help in restoring the fertilization stage. So therefore, usually occurs in the gametes of the organism, and meiosis occurs especially during the gametogenesis, that is during the formation of the gamete, that is the male and the female gamete in plants and in animals, leading to the formation of the haploid gametes. I told you, I gave you the example of the sperm and the ova and also the pollen grain. So therefore, for all these to form that is during gametogenesis, this meiosis type of cell division or the reduction division actually occurs. And some of the key features of meiosis are, meiosis actually takes place in two cycles, wherein it will undergo meiosis 1 and meiosis 2. So, two cycles will occur, but whereas in mitosis, it was only one cycle of division. It will get completed, then it will again enter into the same cycle. But in this case, two cycles, that is meiosis 1 should occur, then meiosis 2 should occur. After that, again, it will enter into the meiosis 1, meiosis 2, again the cycle continues like that. So, therefore, it involves two sequential cycles of nuclear and cell division, that which is called as meiosis 1 and meiosis 2, but only a single cycle of DNA replication will take place. Why single cycle of DNA replication will take place is because once meiosis 1 is completed, there is an intermediate stage called as interkinesis. Don't think Like how in mitosis, it started with the interphase stage, then the mitotic cell division occurred, again it went back to the interphase, again to the mitotic division. Like that, it will not occur here. Here, once meiosis 1 is completed, it will be in the interphase stage, then it will enter into meiosis 1, and once it enters into the meiosis 1 and it completes the meiosis 1, it will not go back to the interphase stage again. It will be in a stage called as interkinesis, and after interkinesis, which is very short, and soon after the interkinesis stage is over, it will enter into meiosis 2, and then it will enter into the interphase stage. So that is how it occurs here. So meiosis 1, so that is why after meiosis 1, DNA replication will not occur. Why? Because DNA replication takes place in the S phase of the interphase, right? So here DNA replication will not occur after meiosis 1. Why? Because soon after meiosis 1, it will enter into the interkinesis stage, and then it will enter into the meiosis 2, and then it will enter into the interphase stage, and then again meiosis 1, interkinesis, meiosis 2 and interphase. Like that it will continue. So that is why there is only a single cycle of DNA replication. Though under meiosis, 2 cycles of cell division will take place, but the DNA replication is only one, because it will enter into the interphase stage only once here. And meiosis 1 is initiated after the parental chromosomes have replicated. So once the parental chromosomes replicates, then it will get, the meiosis 1 will start to introduce, or it will start to get initiated, or it will get started. So here, when the parental chromosomes have replicated, they will start to produce identical sister chromatids at the S phase. We all know that in the interphase, that is the S phase of the interphase, the synthesis of the DNA will take place. So the DNA has to enter into the daughter cells, right? So the DNA has to get synthesized. So during the process of synthesis, what happens? DNA replication will occur, and this DNA replication will produce identical sister chromatids. So all these occurs in the interphase, and after all this, the meiosis 1 will get initiated. And meiosis usually involves the pairing of the homologous chromosomes and the recombination between the non-sister chromatids of the homologous chromosomes. What it means? So we have to understand the chromosomes. First, the chromosomes, they are single. Then the pairing of the chromosomes will occur. So if there are 4 chromosomal arms like this, the homologous chromosomes pairing will occur. This is the homologous chromosome, this is the homologous chromosome. They will pair. And you know that how does the pairing occur? It is because of the centromeres. So the centromeres will have the kinetochores. So therefore, each centromere will, the pairing will occur. Now these are the homologous chromosomes that is formed. Now these homologous chromosomes are called as the sister chromatids. So these are the sister chromatids. And this is one sister chromatid. Now these, which are the non-sister chromatids here? These two are the non-sister chromatids. These two are the non-sister chromatids. So, sister chromatids are the arms of the same chromosome, but non-sister chromatids are arms of 2 different chromosomes. So, here what happens? It involves the pairing of homologous chromosomes and also the recombination. How recombination will occur? Say for example, 2 chromosomes are here like this. So, what did I tell? These are the sister chromatids. What are chromatids? These are chromosomal arms, right? And these 2 that are there, these 2, they are the non-sister chromatids. Now, pairing will or recombination will occur between 2 non-sister chromatids, which is called as synapses. Synapses is nothing but crossing over. How will it occur? Here, I'll just shade it so that it will be easily understood. So, these are 2 homologous chromosomes. Now, if synapses or crossing over or recombining, if recombining has to occur, then here the sister chromatid. So here one more sister chromatid. The arm of this sister chromatid will pair with this. It will cross with this. So here can you see here the non-sister chromatids, one of the arms of the non-sister chromatids crossing over is taking place. Synapsis is taking place. So when synapsis occurs, what happens? So this is, I'm shading it for easy understanding. So what happens next? A little bit of the non-sister chromatid chromosome will get exchanged here. Here recombination is occurring, recombining. A portion of the genes that were present here will get transferred to the neighboring chromosome or where the synapses has occurred. So therefore this is nothing but recombining. So recombining combination will also occur in the meiosis. So these are some of the key features. That is the recombination between non-sister chromatids of homologous chromosomes will occur and 4 haploid cells are formed at the end of the meiosis too. So this is also one important feature. How 4 haploid cells will get formed? First, meiosis 1, the parental cell will divide to form 2 haploid cells. In meiosis 2, again the 2 haploid cells which are now the parental cell, they will undergo meiosis 2 division to form 4 haploid cells. So therefore at the end of the meiosis 2, the 4 haploid daughter cells will get formed and the division will continue as such. The 4 haploid daughter cells will form which will remain in the interphase stage for a longer duration. Then it will enter into the meiosis 1. Once it completes the meiosis 1, it will enter into interkinesis. Then it will and soon within span of time, it will enter into the meiosis 2 and again interphase stage. Like this, the cell division will continue. So this is about meiosis. So next, talking about how these meiosis division occurs. So to easily understand, the meiosis division occurs in different phases. So the main important stages of meiosis division are meiosis 1 and meiosis 2. Under meiosis 1, it enters into prophase 1, metaphase 1, anaphase 1, telophase 1. And in meiosis 1, the prophase 1 is the longest phase. Why? Because if it has to complete the prophase 1 and enter into the metaphase 1, under prophase 1 itself, it has to undergo into various stages of cell division, which is, it is leptotene, it is zygotene, packetine, diplotene, diakinesis. Like this different stages it has to finish and then it will enter into the metaphase 1. But under meiosis 1, the prophase 2 is not that laborious or not that time consuming when compared to the prophase 1 of meiosis 1. Why? Because all the other stages are not there. It will just undergo prophase 2, then it will enter metaphase 2, anaphase 2 and telophase 2. And finally, the division. So therefore, these are the different phases of the cell division that occurs under the meiosis 1 and the meiosis 2. So each of these we shall discuss in detail now. So first, talking about meiosis 1. So I told you meiosis 1, they enter into the prophase 1 stage. So here prophase 1 stage, what happens? The nuclear membrane, they completely get disintegrated, they will be lost and the cell will prepare itself for the further cell division stage, right? So here under prophase 1, you can see the leptotene stage, the zygotene, the packetine and the diplotene, or which is also called as the diakinesis. So in prophase 1, the first stage, that is leptotene, you can see the chromosomes, they come near each other, that is the homologous chromosome. So this is one homologous chromosome. Homologous chromosome. What are they having? Sister chromatids. So this is one more homologous chromosome. So two homologous chromosomes which are similar, they start to pair, they just come near to each other in the leptotene stage. And in the zygotene stage, actually there is the pairing of the homologous chromosome will take place. So you can see here the pairing of the homologous chromosomes will occur. So when pairing of the homologous chromosome occurs, it forms a synaptonemal complex. So what is the synaptonemal complex? Is when chromosomes start to pair, in between the chromosomes there is a bridge-like structure that is formed. So bridge-like structures will form. Just like how railway track will look like, just like that, the bridge-like structures will form, which is called as the synaptonemal complex. So once synaptonemal complex, if it has to be formed or the pairing of the chromosome will occur, during this stage itself, synapses and all that will start. So what is synapses? I told you, two sister chromatids. So to further understand, I'll just draw. So now there are two sister chromatids. So here two sister chromatids. So here there is This is one homologous chromosome. This is one homologous chromosomes which have paired. So here these now synapses should occur or recombination should occur. How will recombination occur? So I told you, I'll just shade this for easy understanding. So pairing or the synapses will occur between non-sister chromatids. So these are the non-sister chromatids. So here what happens is a crossing over will occur. So you can clearly see here the crossing over occurring between the non-sister chromatids. So here, so crossing over will occur. So when crossing over occurs, what happens? There it results in the formation of recombination. So the genes get exchanged like this. So this gene that was present in this particular non-sister chromosomal arm, it got transferred here, and from here it got transferred here because of synapses. So all these recombination process and all that will occur in the prophase stage itself. So here, after the formation of the synaptic complex in the zygotine stage, it will enter into the pachytene stage. In the pachytene stage, now the chromosome forms a tetrad. As you can see, the chromatid arms, one chromatid arm, the second one, third one, fourth one, they will form a tetrad chromosome, and now it is called as the bivalent. Why? Because two chromosomes have combined with each other. They'll form a tetrad chromosome, or it is also called as the bivalent. So bivalent stage will occur, then it will enter into the diplotin or the diakinesis. So in the diplotin, what happens here is the synapses and all that will occur, right? So those chromosomal arms which have crossed over, they will get separated. Can you see here? It looks like an X. Now, after the formation of all these, it will enter into the metaphase 1. So, in the metaphase 1, again what happens? As we know, there is formation of the centrosome, then the spindle fibers, and the chromosomes condense. So, you can see the chromosome condense, and you can see the kinetochores. So kinetochores actually help in the attachment of the chromosomes to the spindle fibers. So this is the metaphase 1, and you can see the chromosomes arranged in one plane on the metaphasic plate. So this occurs in the metaphase stage. After the metaphase stage, what happens? It is just similar to the mitotic cell division that we studied. Here again what happens? The chromosomes, the chromatids get separated because of the division of the centromere. So these centromeres that hold the chromosome. So what are centromeres? The chromosomes, two chromosomal arms are held together by centromeres. So when the centromeres divide, what happens? So when the centromeres, they divide, the chromosomal arms will separate. So they will separate and they will start moving along the spindle fiber towards the opposite poles of the centrosome. So they will start moving towards the opposite poles, and next they will enter into the telophase stage. So in the telophase stage here, actually the cell division will occur, wherein the chromatids will get distributed, that is the chromosomes will get distributed to two of the cells, and you can see the spindle fibers and all that would have vanished. The nuclear membrane will start to develop, nuclear membrane, and the cytoplasm is exchanged between the two cells, and you can see the centrosome. Centrosome is also visible. So this is what occurs in the telophase 1 stage. It is same like that of the mitotic phase. After this telophase 1 stage is completed, it means that meiosis 1 has completed and the cells have divided, having their haploid number of chromosomes. But don't think that it will enter into the interphase stage. No, it will remain in a stage called as the interkinesis. Soon after it enters into the interkinesis, the very next movement, it will enter into the meiosis 2 stage of the cell division. So in meiosis 2 stage of the cell division, here two daughter cells are there formed, right? If you remember, two daughter cells were formed. So these two daughter cells will again enter into the first stage of the cell division in meiosis 2, that is prophase 2. So in prophase 2 again what happens? The same thing. The cell will prepare itself to enter into the metaphase 2 of cell division, wherein it will form the centrosomes, the spindle fibers, then the kinetochores will attach the chromosomes to the spindle fibers. All that will be ready. The chromosome condensation will take place. Everything will get ready as such. It took place in the mitotic cell division. And again here, two cells, again they are dividing. So here also the cell also the same thing will occur. Then it will enter into the metaphase 2. So metaphase 2, again the chromosomes will get arranged in the metaphasic plate. So on one linear line on the metaphasic plate, the chromosomes will get arranged. After that, it will enter into the anaphase stage. So in the anaphase stage, what happens? In the centromere, the division will occur in the centromere. Therefore, the chromatids or the chromosomal arms will start moving towards the through the spindle fibers, and it will locate itself at the two poles of the cell. After that, again, it will enter into the telophase stage, wherein the cell division will occur. So when the cell division occurs, what happens? The two daughter cells will form. So two daughter cells would started undergoing division. So therefore, at the end, it will result in 4 daughter cells being formed. So this is the telophase stage. So next, moving on to the significance of meiosis. So mitosis, cell division, and meiosis, cell division, the process is one and the same. But the only thing is, in mitosis, there was only the prophase, the metaphase, anaphase, and the telophase, and then the interphase. But in meiosis, the cell cycle is very long. Why? Because first it entered into the interphase, then came the prophase, that is the prophase 1. Under prophase 1, it had to undergo different stages of cell division. Metaphase 1, the anaphase 1, the telophase 1, and then it entered into a stage called as interkinesis, and then it entered into the meiosis 2, wherein again it underwent the daughter cells that formed, again underwent the division to enter into different cycles called as the prophase 2, then the metaphase 2, the anaphase 2, and finally the telophase 2, resulting in 4 haploid cells. So therefore, the duration of cell division is longer when compared to the mitotic cell division. And here there is haploid set of chromosomes. There were diploid number of chromosomes. That is the only difference. But other than that, the cell division and all that is almost the same, or the separation of the chromosomes, all that is one and the same. So we'll start with talking about the significance or the importance of meiosis. Here, meiosis help in conservation of the chromosome number of each of the species. That is, any number of generations the cell division occurs, it will maintain the chromosome number in the species. That is especially in sexually reproducing organisms. So here, though it results in the reduction of the chromosome number by half, the genetic material will be as such. The genetic material will not get divided. Only the chromosome number will be reduced to half. Always remember that. So because of that, the maintenance of the genetic material throughout the generation will be as such, and it increases the genetic variability. Why genetic variability occurs in this case is because of the crossing over or the recombination that occurs. You know that we have some of the characters from our parents and some of our own characters which our parents are not having. Why do we have that genes which our parents do not have? So it's easy to understand. So what happens? So when the that is after fertilization, after the that is zygote formation, the fetus will develop, the baby will develop, and all that. So the baby will have some of the characters, or baby will carry the genes from the parents, as well as the baby will have its own set of genes. How will the own set of genes be produced? It is because of the recombination that is occurring. So therefore, recombination occurs in meiosis. We had studied, right, in the meiosis stage, how the synapses or the crossing over occurs, resulting in chiasmata formation and all that. During those times, what happens? The recombination of the genes will occur. So between non-sister chromatids. And because of that, meiosis usually results in genetic variability in the population. That is, when we compare one organism to other, there is some variability in the organisms. Why? Because of the recombination process that is occurring. So therefore, meiosis results in genetic variability in the, that is from one generation to other, there is genetic variability in the population occurring. So variations, and we know that variations are very, very important for the process of evolution. Otherwise, how we would have evolved from the primates, right? So therefore, evolution process to occur, recombination is very, very important, which is seen during the meiosis. So other forms of cell division where mitosis occurs, these variability will not be seen. But since meiosis occurs in the reproductive cells that are such as the gametes, that is why recombination will occur, therefore leading to genetic variability. So this is the importance of meiosis. So I hope you understood the importance of meiosis and the different stages of meiosis, and there is a difference. So the process how the cell division occurs is one and the same, but in mitosis all these recombination, all that will not be seen, but in meiosis there is recombination, chiasmata formation, all that can be clearly visible in the meiotic stage of the cell division. So therefore, meiosis is very, very important for the variation in the genes to occur, so that it will help in the process of evolution. So this was about the chapter that is cell cycle and cell division, wherein you learned about the cell cycle, different stages, that is the interphase and the M phase. Then after studying that, we learned about the meiosis stage in this particular session. So with this ends the chapter, that is cell cycle and cell division. So we shall meet again in the next sessions, wherein we'll start discussing with a new chapter. Thank you.